session of the 3-piece seminar. This is brought to you by a collaboration of the Van Andel Institute, Cure Parkinson Trust, and World Parkinson Coalition. My name is Sara Saez. I'm a research fellow at the National Institute on Aging at NIH, and today's topic is amyotrophic lateral sclerosis and frontotemporal dementia. I know these are not very recurrent topics for the 3-piece seminar, but we wanted to bring awareness of these two diseases that share clinical, neuropathological, and also genetic etiology. The two speakers of today will focus more on the genetic and the transcriptomic part of these diseases. And they will be happy to answer any question that you have. So please don't forget to type your question during the presentation in the box that you can see of Q&A at the bottom of your screen. So the first speaker will be Jack Humphrey. He's a postdoctoral fellow at Icahn School of Medicine at Mount Sinai in New York. And he's going to talk about the integration of transcriptomic data with genetic data of ALS patients across different brain tissues. So when you're ready, Jack, all yours.
Thank you very much for that, Sarah. And thanks for everyone. Thanks to the Cure Parkinson's Trust for inviting me to talk today. Okay, so I hope everyone can see my screen. Today I'm talking about my project, as part of my postdoc at Mount Sinai, where I'm integrating genetic and transcriptomic data for ALS patients across multiple brain tissues. And so brief introduction to ALS, Amyotrophic Lateral Sclerosis. It's a rare progressive neurodegenerative disease caused by the degeneration of motor neurons in the motor cortex and spinal cord, the upper and lower motor neurons. And on the pathology, the majority of patients have TDP43 protein aggregates. So akin in Parkinson's, you have alpha-synuclein in Lewy bodies. The pathological protein we think is involved is TDP43, which is an RNA binding protein. And on the genetics, around 10% of patients have a family history. And this has been used to great success to find genes with rare variants that cause a sort of Mendelian form of ALS. And C9orf72 is the most common of these in patients. About half of all familial ALS patients have a C9orf mutation. And looking across the around 30 genes we have now, there are pathways highlighted, including RNA metabolism, autophagy, and microtubule stability. Now, on the common variant space, we only have about 6 loci from genome-wide association studies. And it's really a black box of how these variants may alter your risk of getting the disease. So I just want to introduce some the data set that I'm working with, which is the New York Genome Center ALS Consortium. So this is a very large multi-center collaboration where hospitals and research institutes across America and Europe have contributed blood and tissue from post-mortem ALS patients. And the sequencing is then performed at the New York Genome Center in downtown Manhattan. So the patients we have are mostly sporadic ALS, or we have some familial cases, C9 and SOD1, a few frontotemporal dementia, and non-neurological controls. And so the current data that I've been working with, they've been collecting RNA-seq across multiple tissues. And we now have 1,900 samples in 11 different tissues. And each donor contributes between 3 and 5 tissues, and we have whole genome sequencing on 513 donors. So this is a wonderful resource to match together the genetics and the transcriptomics in ALS. And so I want to sort of discuss first some questions that we've been thinking about, how we can answer with this data. And so in the transcriptomic space, it's really thinking about cause versus consequence. Can we distinguish transcriptional changes that are caused by the disease itself, the degeneration of motor neurons and TDP43 pathology, and we can distinguish those from those that occur in response to this. So thinking about inflammatory changes and neuroimmune interactions when we look at RNA-seq from what is bulk tissue. And if the first part is true, can we find biomarkers that are specific to pathology? So I'll show some work on these two questions. And then in the genetic space, really think about sporadic disease, because as I said before, a lot of work has gone into familial ALS and identifying rare mutations, but 90% of patients do not have any of these mutations that are known. And what we think is going on is there is a polygenic component to this, where common variations contribute to your risk. And so can we find more of these variants in the data we have? And can we try and think about mechanisms for how they might alter your disease risk? So I've got three short vignettes about the work we've been doing that tries to answer these questions. So the first is on differential expression in ALS tissues. And so as a background to this is we're thinking about bulk ALS tissue, pieces of tissue from post-mortem brain, what we might try and find. Well, according to the literature, the RNA-seq studies that have gone before, where you measure gene expression in post-mortem tissue, you expect to see a reduction in neuronal genes and an increase in glial genes. And we think this is due to glial activation, this idea from this diagram here that there is this interplay between degenerating motor neurons and then microglia and astrocytes that react to factors released when cells start to degenerate, which become then active, they proliferate, they change their states, and there's also this other source of immune cells from the blood vessels, so T cells and monocytes that infiltrate in. And so this is kind of what we expected to see going in. So what I put together is the 8 tissues here where we have sufficient sample size to do this in. These are the numbers of controls and for ALS cases. Here I'm lumping sporadic and familial cases together to get the largest sample size we can. And when you perform a differential expression, which is you take each gene and you compare the expression of that gene in ALS to control to find genes that go up more so in ALS or down more so in ALS, these are the results we find. And what you can see is, though, if you take all the genes without looking at and just taking a nominal cutoff of 5% p-value for each test, you see we have a lot of genes changing in each tissue. But when we correct for multiple testing, which you should do, and some of the previous studies didn't really look at this so well, is actually if you're careful, the only changes you can see are in the spinal cord, specifically the cervical and lumbar spinal cord here. You can't identify any significant differential expression at 5% FDR in any other tissue. And so this was initially a little disappointing. We thought, well, what does it mean to have all these frontal cortexes, cerebellums, and things like that? Can we learn something from the other tissues, knowing that we do have signal in the spinal cord? And so the first thing I did was to correlate the expression of each gene, the direction that it goes in ALS versus control across the tissues to correlate these things. And what you find is that the spinal cords all have correlated directions of effect. You can see that the cervical and lumbar spinal cord are very highly correlated with each other. And the thoracic spinal cord, even though we didn't find any differentially expressed genes at 5%, you can see they correlate. So you can see between the two spinal cord tissues here, we have a nice correlation. And then when you look at some of the other tissues, what stands out is that the lateral motor cortex here that we're sequencing has a positive correlation, and it's higher than the other tissues. So what it suggests is that the changes we find in spinal cord are reproduced in the motor cortex, although the effect sizes are different. They're smaller, they're harder to find, and this could be due to the sample size. This could also be due to heterogeneity, as we know that ALS is a heterogeneous disease, and it may be harder to identify when you group all the ALS samples together strong changes in the other tissues. And so because we see these potentially sub-threshold changes occurring, that in the motor cortex, for example, there are shifts that correlate with the spinal cord, we applied a technique called gene set enrichment analysis. And the basic idea is you take your genes that go up and down, and you take a set of genes that you know, for instance, are markers of a particular cell type, and you can test whether those genes are more upregulated than downregulated. So in this toy example here, you can see the gene set happens to be in genes up here, and it's depleted down here. So you can generate a p-value and enrichment score. And so by applying gene sets and marker genes from different cortical cell types, we can estimate sort of the shifts in cell type composition in the data sets, in the tissues. And so what we see in the 3 spinal cord tissues is you get a strong downregulation of oligodendrocytes, which has not really been observed before. And it's throughout all 3 tissues here and in the lateral motor cortex. And then you don't see a strong signal in neurons. What we expected to see was a downregulation of neurons. In fact, you see actually a small upregulation in the thoracic spinal cord, but it's only significant here. And you see when it's gray, that means this didn't pass multiple testing. But you see microglia, very strong enrichment here. And the lateral motor cortex seems to resemble the spinal cord, whereas the medial motor cortex, this other section of motor cortex, does not. And so I think preliminary preliminary work going back to the pathologists to work out exactly what they mean here is the lateral motor cortex is most likely the primary motor cortex where the upper motor neurons actually are. And the medial motor cortex is most likely the supplementary motor area, which may be affected, but it's more distal from where the cells are actually dying. And you see in the medial motor cortex, you have this downregulation of endothelial cells and astrocytes here. So because we have this glial signature here, this microglial activation, we're interested in this increase in microglia perhaps is just a shift in, there's more microglia, or is it a shift towards activation? And so for that, we took gene sets that are known, genes that respond, that are in microglia that respond to inflammatory stimuli. And we see this general increase in the spinal cord and the motor cortices, and also in astrocytes. So some other gene sets from astrocyte activation also are increased here. So just to summarize this work so far is that in bulk brain sequencing, what we think is happening, we're mostly picking up cell type composition differences. You know, there's differences in microglia and astrocytes, and there's a big change in oligodendrocytes, but we can't really see neuronal changes. You know, it's hard to find pathology specific changes. So we think that ALS is a disease of motor neuron degeneration. It's very hard to see that in the bulk RNA-seq that we have from tissues. And so moving forward, we want to generate single cell data where we should be able to then assign cells to particular cell types and actually see, is this a composition change? Is this a state change? So that's that project. Next, I want to talk very briefly about trying to find markers of pathology. So actual TDP43 pathology in the cells that have it in the bulk brain, what's actually going on. And STAthmin2, I think, is a really nice potential biomarker for TDP depletion. And so STAthmin2 is a gene involved in axonal development. And in the structure of the gene itself, within an intron, there is an intronic sequence that contains a binding motif for TDP43, which is, because it's an RNA binding protein, the protein will bind RNA and influence splicing. And so what we think is going on is that when TDP forms these pathological protein aggregates in the cytoplasm, it leaves the nucleus. So it can no longer bind RNA as splicing occurs in the nucleus. And what happens is this exon is then included and forms this truncated STAthmin2. And so in experiments with Len Petruccelli's group, we show that we can knock down TDP43, which leads to a reduction in STAthmin2, but an increase in the cryptic exon. So the full length version goes down, but the cryptic exon goes up. And so this was in cell lines. I'd say this has been published before. This is mostly looked at in knockdown data. And we wanted to apply our big catalog of patient tissue to this question. Can we see this cryptic exon, this exon here, and does it segregate by disease and by tissue? And the answer is yes. So I developed a computational assay to find this in RNA sequencing data. Essentially, you have the reads covering the normal transcript, and you have the reads covering the cryptic exon here, and you look for reads that splits across here. And what you can see is that the presence of these reads is very specific for TDP43 ALS. In cases with SOD1 mutations, they don't have TDP43 pathology, and we don't see it, and we don't see it in controls either. So it's highly TDP specific. And when we look across tissues, it's occurring in the tissues that we think TDP pathology actually is there, right? So we know the cerebellum is not affected in ALS, or the occipital cortex. So we see the spinal cord is very well represented, as is the motor cortex, and a little bit the frontal and temporal, but less so. And we think this is generally ALS patients with pure ALS do not have pathology in the frontal and temporal cortex. But when we look at patients that have cognitive symptoms where they have defined frontotemporal dementia, we do see more STAthmin2. The drawback to all this is actually, this has been published, but this is deep in the supplementary, is the number of reads you actually detect. If you think we have maybe 60 million RNA-seq reads per sample, maybe 10 of those reads will correspond to this TDP pathology biomarker. So it's a very small number. And I think going in, we thought we'd see this more often. You know, based on the knockdown data, it's very strong and obvious, but actually in a patient brain, in a patient spinal cord or motor cortex, it's a very weak signal. And so I think this is because the TDP pathology is actually affecting a very small minority of cells, and pathology data backs this up in the spinal cord. You know, the number of affected neurons, or the neurons that are still there to have TDP pathology because someone had died, is actually very small. And so using this data to find more biomarkers specific to TDP pathology is going to be very difficult without something like cell sorting or single cell. So finally, I want to talk about genetics. And so using common variants to associate with expression and splicing in tissues to find new ALS risk genes. And the approach we use for this is a schematic from a recent review on functional genomics, which is very, very helpful. This is an approach called quantitative trait locus mapping. And so this identifies common genetic variants that have an impact on gene expression and splicing. And the way you do this is you have your, say, gene expression here, your phenotype in your tissues, in your patients, so in your individuals, and you have the genotype. And we take the whole genome sequencing and look for common genetic variants. And then we perform linear regression, like so, associating genotype with gene expression. And what we end up with is we do millions of tests for all the common variants that are close to the gene. So we're looking for cis genetic regulation or genetic variants close to within a megabase of each gene. And this gives us a lot of associations, some of which will pass a threshold. So here's our lead variant. And then you have these other variants down here. And the reason you see this kind of peak of association is because of a phenomenon called linkage disequilibrium, where individual genetic variants are not independent from one another. In fact, genetic variants that are close by are often co-inherited because of the way recombination works. You have variants that will be co-inherited. So I performed this analysis across 7 different tissues. Like so, we have, this is using all the samples together, not just the ALS, to get as much power as possible. And then these are the numbers of genes we can find with a cisgenetic QTL, either affecting expression or splicing. And for splicing, just to let you know, the regression we fit is an association with the ratio of splicing for a particular intron. And so we find generally that the more donors you have, the more genes you can find. It's just because of power. And generally you find fewer splicing effects than expression effects. And because it's harder to work with splicing than expression, you have fewer reads to work with, and you have less power. So what do we do once we have this? Well, the next step in our functional genomics workflow is to perform a technique called co-localization, where you take your eQTL analysis, your associations like this, and then you compare it with a GWAS, a genome-wide association study. And so in ALS, for example, this is, it's the same X and Y axis, it's genomic position by p-value, but here the p-value is for the test of, does this SNP alter your risk of ALS? Is it more frequent in ALS cases than controls, or less frequent in ALS cases than controls? And again, you'll have a lead variant and then some other variants down here that are all in linkage disequilibrium with it. And so you combine these 2 associations and you have multiple hypotheses that you're testing. And the hypothesis we're interested in is, are these 2 effects generated by the same variant? Is it the same variant that affects your cis expression or splicing of a gene and also alters your risk of ALS? And so if they don't co-localize, you see the distributions are separated, but if they do, they should be very similar, they should overlap. So what I did is I took the most recent ALS GWAS, which has 6 loci here, and I reduced the significance threshold a little bit to start seeing if we have some sub-threshold hits that might be interesting, that perhaps it didn't quite reach your cutoff genome-wide significance, but with some co-localization, it gives you a bit more evidence to consider it a risk gene for ALS. And so this is how you plot the results. This is the triangles of the splicing QTLs here, and this is the probability of it being a co-localization. So the higher probability is better. And you can see that of our 6 genome-wide loci, 2 of them have a QTL in at least 1 tissue. And for C9orf72 and UNC13A, it's all splicing QTLs. And I can probably talk about this in the discussion, but I want to highlight some new suggestive loci that I think are also interesting. So this is when you go into suggestive hits, you lower your threshold a bit, and you can see we're picking up several genes now that have both eQTLs and splicing QTLs, expression and splicing, that may be from the same variant that causes ALS risk. And the gene I want to highlight is ataxin-3 here, where you can see that in the cerebellum and spinal cord and motor cortex, you're seeing some evidence of co-localization with a splicing QTL. So why do I think ataxin-3 is interesting? Well, I think it's a good candidate because it's the gene family it's in, the ataxins, are known to cause a motor disorder called spinocerebellar ataxia. And what's interesting about the ataxin-3 ataxia, which is called ataxia type 3, or SCA3, or Machado-Joseph disease, is that these patients have TDP pathology in their lower motor neurons. So there's already a pathological connection with ALS. But on the genetic side, the other family members, ataxin-2, ataxin-1, and ataxin-8, have all been associated with ALS risk. And this is because the ataxin genes contain glycine repeats, these CAG repeats. Most people normally have between 10 and 30 in ataxin-3. And if you have a large number of repeats, this can cause the ataxia. But for these other genes, it's been found that intermediate numbers of repeats also alter your ALS risk. They increase your risk of getting ALS. And so with the very little time I have left, I just want to show you some sort of fine mapping work we've been doing on the ataxin-3 locus to see where the splicing QTL actually is and how this interacts with the repeat. So this is a plot of the ataxin-3 transcripts. They go from right to left here. You see this long transcripts and little short transcripts here. You have a polyadenylation site here and here. And what you can see is that when we overlay the introns that are actually associated, because this is a splicing QTL, the introns that are associated with ALS, because they're co-localized, are all in the 3' end here. The GWAS SNP is here, the top one, and then the top QTL SNPs are here. And when we look at the linkage disequilibrium between this QTL SNP and this GWAS SNP, they're in perfect LB. So it suggests that actually there's a single causal variant that's affecting both ALS risk and splicing of this part of the ataxin-3 gene. Now, when we look at where the expansion site is, it's here in exon 10, just next to where the splicing occurs. So this suggests that actually the GWAS SNP is tagging the expansion to some extent. So the SNP correlates with splicing, the ratio of intron usage, but it also correlates with the expansion length. That actually, if you don't have this variance, if you're homozygous for the reference, you just have a range of expansion lengths. But people that have this this variant, it's restricting the expansion to a higher set of lengths. And we did this by quantifying with Expansion Hunter on the whole genome sequencing. And I discussed this further in the questions. So I'll wrap up, just summarizing what we've done. And so, you know, in bulk brain, cell type composition is probably the dominant signal that we're getting. And looking for direct pathology-related RNA phenotypes is going to be very hard with this data. And so I think this is why we need to push for single cell or spatial transcriptomics to better study this. But I think the real use of the data is connecting the genetics, because it's infeasible to do single cell, you know, on 200 patients. But with the bulk data, we can find these expressions, splicing QTLs, and really link them to ALS risk. Although I'd say for splicing QTLs, it's going to be good to have the full transcripts. And so we're looking into long read RNA sequencing to see that, to really identify the disease relevant transcript. So with that, I'd like to thank everyone at the Raj Lab who are hiring postdocs, I should say. My old mentor, Pietro Fratta at University College London, the Genome Center, Len and Mercedes at the Mayo Clinic, everyone that contributed data, and of course the ALS patients and their families that donated this really precious resource. So here's my email and my Twitter handle, and I'm done. Thank you very much.
Okay, thank you very much, Jack. It was very interesting. And I have a few questions myself, because for the data that you present, I think we talked about this before, but did you already apply polygenic risk score, kind of relate the changes that you see with your expression and any kind of burden of genetic risk that this cell type could carry? And I'm asking this question because for the project that I did, I only used polygenic risk score. And by using polygenic risk score with ALS patient, we nominate oligodendrocytes. This is one of the cell types that pop up as these cells carry a high burden of polygenic risk variants. But also we found interneurons. So you don't find neurons. Do you think this is because, you know, like still the sample size is small, or because you think like maybe the changes, the genetic changes that can happen affect other cell types instead of being in their cell type?
Thank you. That's a really good question. The short answer is no, I haven't done that analysis. The slightly longer answer is I would like to, and we're going to do it at some point. I think what you're highlighting is there is a disconnect between what the genetics will tell you in terms of the cell types that are implicated in the risk of disease, and what the bulk transcriptomic data will show us, which has shown me, right, is that you're mostly picking up consequences rather than causes. And so I think the sort of orthogonal approach to your PRS would be to take these co-localizations that we now have with risk genes in our tissues and say, well, are these co-localized, are these affecting genetic variants that are in cell type specific regulatory regions, right? What would be great to say is, oh, ataxin-3, for example, is highly expressed in oligodendrocytes, and somehow, you know, the variant is only affecting a transcript that's in oligodendrocytes. And so we have a project that we've been doing this in Alzheimer's disease where we look at microglia, for example, because there's a lot of work that microglia are the causal cell type in one of the causal cell types in Alzheimer's disease. And I think a big push for the whole field, right, it would be great to be able to identify causal cell types through these genetic methods that we're both applying.
Okay, so there is a question. I mean, you have a couple of questions. So the first question said, hey, Jack, very interesting. There is a paper in bioRxiv that identifies using polygenic risk score that oligodendrocytes have enrichment of genes that increase the risk for ALS. Do you think there is correlation with your data? And I think you already replied this question.
I wonder whose paper that is. Whose paper do you think that is, Simon?
So the next question is like, thanks, Jack. Could you use the same resource for a single cell version of this analysis? And if the RNA is good enough for long read analysis?
So he must be reviewing my grants because these are important questions. So I think the second question is a very important one. So for RNA quality and long read analysis, because it's post-mortem tissue, it's from frozen brains that may have been in the brain bank for over a decade, is that RNA quality is a really thorny issue, especially when you want to do long read RNA sequencing, because the idea would be that you would sequence the entire transcript in a single read. So you need that entire transcript to not have been degraded, because RNA over time will degrade 5 to 3 from the 5' end to 3' end. And we know from published PacBio data, right, for example, that we see a lot of this fragmentation. So I think going forward, it's going to be difficult to use the post-mortem data we currently have. And I think it might be better to look at fresh brains. So for the microglia project, for example, we isolated microglia straight away from at autopsy, which really helps keep your quality high. Now for the single cell analysis, I would love to do a single cell version of this analysis. I think the biggest hurdle here is cost. I think this is coming. A lot of groups are doing, especially in the Alzheimer's field, probably in Parkinson's too, doing patient level single cell analysis. And this would be fantastic, right? If you could reproduce this in enough samples, I think cost is the main barrier to that right now, but the cost is going down all the time.
Okay, and I have another question. So from the genes that you identify, like this, using colocalization analysis, so SSFD1 and ACSL5. So do you think those genes are like specifically expressed in any of the cell types that you previously observed, like oligodendrocytes or microglia? So these genes could be specific or highly expressed in those cell types?
That would be the hope. That would be the hope, yes. So, I mean, one of the analyses that I tried to do and failed at was doing a cell type interaction QTL analysis. So this would be where you would estimate your proportions of cells in your bulk tissue. So for example, oligodendrocyte proportions. And then you would use that as an interaction term for your QTL. So you would look for genes where the genotype alters gene expression, but it does so more when you have a high proportion of the cell type. And unfortunately, that we don't have the sample size to do that analysis. You can't find anything. I know that GTEx have done that recently, and they do find a small number of hits, but I think that's difficult with the current data set. I think what's going to be more feasible in terms of identifying cell type specific things is using existing catalogs of, say, cell type specific epigenomic data. So cell type specific enhancers, promoters, and that's something we've been working on in the background.
Okay, so you have one last question. Hi, Jack, great work. Can you please elaborate how you genotype the repeat expansion? Also, if I may, a second question, you mentioned about the plans for single cell sequencing. It is possible to apply the splicing quantification pipeline to use your QTL mapping also on the single cell sequencing. You need to be very brief.
Right, okay. So first of all, so Expansion Hunter. We used Expansion Hunter. I think they used the default version. This was done at the New York Genome Center. where Ataxin-3 is a known expansion, so they just ran the normal known expansion hunter. So that's fine. Second question, single cell sequencing, the highest throughput, cheapest single cell we can do right now is 10x 3' sequencing, which means you don't capture the full transcript. So I know there's a lot of work currently to create full transcript coverage, single cell data with SmartSeq, things like that. And that would be ideal to do splicing, right? To do single cell splicing. For expression, it's fine, but for single cell splicing QTLs, we need a bit more time for the technology to catch up to doing full coverage.
Okay, good. So thank you so much, Jack. If anyone have any question, can type the question on Twitter or also in the 3P Facebook page. And Jack, I'm sure he will be happy to answer this.